Okay, ladies and gentlemen, we're going to make a start. Good evening and welcome to you all. It's a pleasure to be spending some time with you tonight. Jason and Tamara have asked for me to talk with you a little bit about ozone. This presentation night is being recorded, so the video will be available through Jason afterwards if you want to come back and watch it again. So let me just share my screen and we'll get going. All right. So the purpose of this presentation tonight is to really give you just a clear understanding about ozonated water, what it is, where it's used, and where it can be used in clinic. I don't have any commercial interests in ozonated water systems, although I've worked with many and also been involved in water treatment. And indeed, I'm currently on a patent application for a water treatment, but it's using a slightly different technology. So I don't have any specific other conflicts of interest to declare to you. So we're going to talk first of all about the science of what the ozone is doing to different sort of microorganisms, be they bacteria or fungi, protozoa, viruses, etc. And then spend some time looking at the issues about how you deliver it in a dental practice in water. And the concept I'm going to take you through is called the TC concept, which means how time and concentration are very much linked together. I will share with you some of our research, some other research from the university as well in ozonated water, so you can get a bit of a sense of where we feel the technology is going at the present time. So to begin at the very basics, ozone is often called triatomic oxygen because it's O3 rather than diatomic regular oxygen, which of course is O2. First identified in 1840, and it has a very, very characteristic odor. And this is actually what gives it its name, which comes from the Greek word ozine, which means scent. However, we can not wait for a lightning storm or electrical discharge to get ozone. We can create it in different ways. So we can create it by electrolyzing water, putting electrical current through water at a voltage above 1.23 volts with a special type of catalyst, for example, a platinum electrode. We can electrolyze water very successfully. We can take oxygen and expose it to UVC light, which is done with special ultraviolet lamps. And we can also basically create controlled lightning type electrical discharges. And several of the ozone systems that I've built use exactly that sort of principle. What all these share in common is they take regular oxygen, they split it into parts, and they recombine it in a way that means that you can get some possibility of reforming into O3 rather than back into O2. However, the O3 is actually quite unstable, and under normal conditions of, for example, room temperature or body temperature, it wants to convert back into regular diatomic oxygen. So unlike other gases like helium or hydrogen or nitrogen or CO2 that you can put in a cylinder and store, you can't do that with ozone. You need to generate it because of this decline. So what each of these graphs show you is the effect of pH. So basically what happens when you elevate the pH, that tends to alter and shorten the half-life of the ozone. And basically the amount that remains drops away. And this has been really well described in the literature. And it's an important concept to understand that when you're going to use ozone to wash your hands or as a mouth rinse or put it into your dental chair or whatever, you need to understand that it has, if you like, a little ticking time clock inside. So if you look at all these graphs that show the concentration in parts per million, you can see how those basically drop down over time and that the amount of reduction varies according to the pH. And you can see that really clearly in all these scenarios here. In fact, if you want ozone in water to be extremely stable, for example, if you want to get several hours out of it, you have to deionize the water. And this is some work that's been done at the University of Queensland using different types of deionizing systems. So the first curve you can see here is just regular tap water. So if you put ozone in tap water, you can see that basically after about 30 minutes, it's more than 90% gone. So it has a typical half-life of somewhere around 15 to 20 minutes at room temperature and regular conditions. If you deionize the water, which is what all these other three alternatives are, you can see that you can get more in, and also that you can actually get a half-life which is somewhere more around about 2.5 hours. And this allows you to do some really clever things with it. For example, put it in a drone and spray it around in order to get environmental disinfection, which is exactly what this particular paper is all about. If you recognize some of these names, or if you don't, you might recognize Paul Young, who's the guy behind the UQ version of the COVID vaccine. So let's talk about what ozone is doing to pathogens. So with bacteria have got cell walls either made out of different layers of lipid if they're gram-negative, or lipoteichoic acid if they're gram-positive. Ozone's really effective at damaging those sorts of membranes, but it also damages the outside of fungi and also viruses. So it's not something which is limited only to bacteria. It'll actually have a go at practically all forms of microbial life. Then you might be thinking, well, wouldn't that be a problem with human cells? Well, human cells have got quite different membranes from all things that I've just mentioned, and we're well endowed with natural defenses against these sorts of molecules because they are encountered in the environment, and not surprisingly, we have a defense system against them. So when you run into bacteria or spores, ozone will inactivate that. If it's regular bacteria just growing in the ordinary environment, what we call vegetative bacteria, those will be killed off. If they're spores, they'll be inactivated, which means they can't germinate. If they're viruses or larger parasites like amoeba or even tapeworms, they'll be wiped out. And this is why ozonation has been used to disinfect water all around the world for well over 100 years. It's used quite a bit in Australia, and if you're in Brisbane, I know a couple of you are, then at the moment you're drinking partially water from the desal plant, the Gold Coast, partially water from the Gold Coast dams, particularly the Hinds Dam, and partly Brisbane sewage, which has been ozonated in order to remove all the normal viruses and bacteria you find in sewage, and that's been blended with those other two water sources, and that's what's coming out of the tap in Brisbane right at the moment. And that's been the case for some period of time. So it's a normal thing to treat water with ozone in order to disinfect it. And that shows you that it's very, very broad acting in terms of its antimicrobial effects. And that's why it was approved in Australia in 2005, officially by the NHMRC, to treat water. And that's why it's used in Sydney and Brisbane and many other municipalities for water treatment. And not surprisingly, what we do in dentistry is like a mini scaled down version of the same type of technology. In fact, this is taken from the current version of the Australian Drinking Water Guidelines, the ADWG. And this really neat table summarizes chlorine versus chloramines versus ozone versus chlorine dioxide versus ultraviolet light. And again, without taking you through the table in its own right, you can see that the rating for antibacterial activity and antiviral activity is actually pretty good. There's no residuals. You only need a short contact time. There is some effects of pH, but it's actually reasonably reliable as a technology in terms of comparing it to other things you're probably familiar with, like chlorine. Now, the aqueous form of ozone has been used to disinfect water for a very, very long time, not only to drink, but also, for example, in commercial settings like restaurants, in livestock, if you've got piggeries or if you've got dairies, in those sorts of environments as well. And of course, in healthcare and medicine for exactly the same reason. So these are just some relevant references around that. All of you have, I'm sure, drunk water from a bottle. And every time you pick up a bottle of water and you crack the seal, you are actually taking water that's been sterilized by ozonation, and that's what you're about to drink. So it's used extensively to treat bottled water, and it has been for many, many years. people now put on their food to get rid of some of the common pathogens that are transmitted on strawberries and lettuce and those sorts of things that obviously grow in the ground, and so there's going to be some pathogens there. But it's also used to break down pesticides and residues, including in industrial settings. So it's actually quite a widely used type of chemical treatment. Here's some examples from Rockpool Restaurant in Sydney, showing some of the underbench water ozonators that are used to basically spray down the seafood and the lettuce and so on in order to keep those things optimally fresh. Now, all this sounds great. It's not actually new. If you trained in microbiology in the 1970s, like I did, it was sort of standard stuff in a way. And here's one of the sort of classic papers from way back from 1975, which is a bit before I started dental school, but just talking about how well the ozone treatment can work on viruses and bacteria and how you can actually modify that. And we went from this paper to do some work about ways of enhancing what ozone does in places like root canals. If you use activation with an ultrasound device, you use a ultrasound activation of a file tip and those sorts of things. Now, when you look at the key pathogens that you're trying to get rid of in normal tap water, if you're going to put that water into your dental chair, there are a couple of key pathogens that you would want to remove. One of those is Pseudomonas aeruginosa. It's one of the organisms that we grow in our lab when we're testing out different antimicrobial strategies because it's hard to kill. It's a fairly tough nut. We also normally test coliforms like E. coli, staphylococci from the skin like Staph aureus, difficult to kill gram-positive bacteria like E. faecalis, a well-known endodontic pathogen, and of course Candida albicans, which is a major pathogen found in the mouth, but often causes systemic illness. So when you look at the problems of water like that, and we've tested lots of different therapies in water treatment, these are the sorts of things that we typically test them on. So what you find is that as you up the concentration of the ozone, the growth reduction alters in proportion to that. And this is this concept of concentration and time. So some organisms are easier to kill than others. So Staph aureus and E. faecalis are both gram-positive, and so their dynamics of killing are actually relatively similar. They have fairly tough outer membranes. When you look at gram-negative bacteria like E. coli and Pseudomonas, the killing effect is slightly different. So when you're looking at how much ozone do you need, you always calculate that based on the toughest target, not on the easiest target. And if you put bacteria under the electron microscope, the scanning electron microscope, here are normal healthy organisms at the start, then what you find is when they've been ozonated, these are the blastoconidia of Candida, these are typical coliforms, and these are the typical little bunches of grapes configurations of Staph aureus. What you see is their entire shape gets distorted, and then everything actually just gets fragmented into bits. So that's what the ozone is actually doing. It's actually tearing these things apart by basically damaging their cell membrane. So I thought this was just a really nice set of sequential pictures just to show you that. Now, obviously, these are the reasons why we use it to treat sewage, and its ability to inactivate everything I've spoken to you about before is really the nub of where it's currently used. And interestingly enough, during the COVID pandemic, I got a couple of questions from people about, would ozonated water do anything to COVID? And I said, well, obviously, because COVID is a medium-sized virus. So difficult to kill viruses are really small. Things like Hep A, Hep E, rotavirus, for example, noroviruses, respiratory syncytial virus, those things are very small, very hard to kill. Very large viruses like adenoviruses or herpes virus are actually pretty easy to kill. And coronaviruses aren't that difficult. And there's now quite a bit of evidence around inactivation of coronaviruses, and obviously the UQ research has been focusing on that very, very much. There's actually a fact sheet on ozonation of water, which is on the ADA COVID website that I wrote about 5 or 6 weeks ago if you're interested, because it has quite a long list of references, that all of which are included in tonight's lecture, by the way. But if you want them all collated nicely, then you might find that a useful resource. So on a virus that it has an envelope that's a big virus, it's basically going to wreck the outside of that envelope. Then it's going to get in and start to wreck the DNA or RNA of the virus. On a non-envelope virus that's small and difficult to kill, these are coated with proteins. It diffuses through the protein coat and then starts to destroy the nucleic acid on the inside. It also wrecks all of the attachment proteins on the outside, which means that the virus can no longer stick onto cells. And this means it's no longer infectious. And this is why I referred to the earlier work about ozonated water in a drone being sprayed around some outside area. It's actually a very effective strategy for exactly that reason. Now, there are lots of other viruses that have been looked at in terms of inactivation. These include things which are very small, such as bacteriophages, which are the viruses that infect bacteria, and obviously the hepatitis and other sorts of viruses. One of the viruses that's a really good test case for how good ozonated water is norovirus. And I'm sure you've all heard about norovirus. Before COVID was spread on cruise boats, norovirus was the reason that people got sick on a cruise boat. Very, very small, very difficult to kill, and very resistant to things like alcohol-based hand gel. And part of the solution for that was to start to use technologies like ozonated water that do actually work extremely well against things like noroviruses. If you leave your dental practice and you go off to work in an aged care facility, then you'll find that one of the problems of going there is that a lot of the residents who live there carry norovirus, or they carry Clostridium difficile. And these are very contagious and they're easily spread by contact. And so ozonated water has been brought into nursing homes to try to reduce that risk, because these can be quite serious and fatal infections in nursing homes. Anyway, if you want to read more about that, this article that I wrote in 2020 talks about the specific problems of C. diff and norovirus with reference to the nursing home literature, and talks about how more than 99% of norovirus, you can inactivate that at 1 part per million within 2 minutes just for ordinary tap water. So something that's not very sophisticated can actually work very well. Now, in reference to SARS-CoV-2, people have tested not only ozone in water, they've also just tested ozone in the air, often generated with a big bank of UVC lights. And there's actually been some really interesting research done on ways of doing that. The main problem in doing it that way is that you can't have any person or animal in the room, because the level of gaseous ozone you generate is at a sufficient level that it's not only irritant, it can also be toxic. So it's basically used for rooms that have got no people or animals in them. In other words, after everyone's gone home, you send the robot in to basically move around with the UVC lights that produces the ozone from the regular room air, and that acts as a type of surface disinfectant. So in terms of aqueous ozone, obviously aqueous ozone will inactivate SARS very quickly, and this is why we talk about being able to use it as a mouth rinse. So for example, there's good evidence that with around 3/4 parts per million, you will inactivate even high loads of SARS-CoV-2 within a couple of minutes. So it's actually quite a susceptible virus in terms of that. And if you want to read up more, this is a great paper published by Paul Young's group. This is in the School of Chemistry and Molecular Biology, who I have a collaborative research interest with. We published quite a few papers in clinical microbiology with this group in the SCMB. So basically they show that the concentration effect, the amount of surviving virus, this is the baseline control when there's no ozone. When you've got 0.3 ppm, when you've got more than 0.75, it's basically all or nothing. So that's a fairly typical thing you see with ozone in water. Once you reach that threshold level of about a ppm, you wipe everything off the deck. So moving on to then, well, how do we implement these sorts of ideas in a dental practice? Well, an obvious thing is to connect some sort of ozonating device to your regular reticulated tap water. Now, in order to connect a device to the tap water system, it requires in Australia and New Zealand a watermark, which is basically an official certification that the device is safe to connect to a regular tap. And of course, a ozone infection control tap, or a KIC tap as it's often referred to, has exactly that specific certification. So that can be installed by a plumber or somebody else to connect to your water system, and then you can have ozonated water on demand from a sensor, or you can have some other type of tap, but the basic ozonator is built underneath the sink. You can also use large units which are basically there to deliver much higher volume. It really just depends on scale. So it can be scaled from a very small flow up to a very large flow. It can also come in a spray bottle and in a self-contained bottle. And if you're interested in those, then perhaps talk to Jason or Tamara afterwards. So into these taps, there's basically two types. There's the tap that you're basically just going to screw into the bench and it's fixed and you point it into the sink, or you want to have a tap that you can actually pull out that's on an extendable hose, like the example on the right-hand side. Now that might be more useful in a sterilizing room sort of environment, whereas normally in an operatory, you'd want a fairly simple tap like a KIC tap, which just simply sits on the bench. And the reason for that is you'd probably want to do other things with the ozonated water in the surgery. For example, to feed it into your dental unit, to put it into, if you've got separate irrigating bottles with your ultrasonic scaler, to put it into that. If you want to use it as a pre-procedural mouth rinse, because you want to offer an option that has basically no taste or no irritation, or you might want to use it as an additional surface disinfectant. So having it as a tap in the surgery, most people find is the most convenient installation type. So if you look at a KIC tap, this is what it looks like in the box. There's basically the actual tap, there's the connections to that, and there's basically the little device which does the ozonation, which you can see here, and obviously some instructions. So just sort of unboxing that a little bit, this box not only generates the ozone, it's also the control box which feeds back to that little passive infrared sensor, which is basically what then activates a solenoid to flow the water through the unit in the first place. So it's a relatively simple type of installation. So you could add that tap to an existing sink. You basically drill a hole, put on the side of your existing tap with a flick mixer, like these couple of examples shown here. That's a very simple and straightforward installation. Alternatively, you might say, well, maybe take the existing tap out and just replace it with one of these. Of course, you'll only get cold water in that case, but if you don't live in a very, very cold climate, then you probably won't be too worried about just having one temperature of water in order to wash your hands. And I can tell you in the building that I work in, which I also designed, there are no hot water taps in any of the about 25 toilets in the building, and they're not used routinely for hand washing because you actually don't need it, at least in the climate of Queensland. Down south would be different. You might certainly want to think about a hot water tap there for washing your hands in winter, but in Queensland, you don't really need it. So in that case, it could actually be just a replacement. And that's what these three installations here are. They're just a direct replacement scenario. Or if your practice has a scrub sink, you can add that on to the existing scrub sink. So you've got that there as well. And you can use that while you're doing your surgical scrub with your antimicrobial soap, if you want to do that. In fact, we'll come back and talk a bit more about this application at the end, because it's actually quite an interesting little thought process around that one. Of course, if you've got it in your operatory, then you can certainly use it for hand hygiene, for your normal hand washing interventions that you would do at the start of the day, at the end of the day, before you go to lunch, and those sorts of things. So can it actually be sufficiently effective when you compare it to things that you know, like alcohol-based hand gel? And this is an interesting recent study which really came out of a hospital in Norway where several of the ophthalmic surgeons, because they'd been doing these lists, and I'm not sure how much time any of you have spent in theatre, but if you've ever watched an ophthalmic list, you know, they punch through patients very, very quickly. You know, in dental, you know, we have much, much longer sessions for patients in theatre, but in ophthalmic surgery, they're much, much faster. And they end up with a lot of surgical hand washing events. So not surprisingly, some of the ophthalmic surgeons get a lot of skin irritation on their hands from just the sheer frequency of exposure to water and to antimicrobial soap. So one of the authors of this paper actually had to interrupt his work. He couldn't do the intensity of work because his skin was breaking down. So it drove them to look at the question, what if you used ozonated water in someone who just couldn't tolerate using alcohol-based hand gel because of incredible stinging? And so this was an interesting little study. So they recruited 30 students from the nursing college associated with the hospital. And they then did the standard EN 1500 test, which is basically the certification test that you do to get approval as an alcohol-based hand gel for use in a clinical facility. So they basically swab onto the skin a strain of coliform, so E. coli bacteria. And then they either put on standard alcohol-based hand gel made out of ethanol with isopropanol, or they put on ozonated water at either 0.8 or 4 parts per million. And they used both of those for 30 seconds. And it was a crossover study, which meant that some of the students used one protocol and then switched over to the other, and vice versa. So they could then track the efficacy in individual people. And that turned out to be a really, really neat way of doing the analysis. It's not new. I was involved in research into alcohol-based hand gel when I was a postdoc in North America. I spent some time working in a medical school at the University of Pennsylvania. And we used to do these large studies where you'd put a massive handprint into a giant culture dish and count all the colonies. And you would design the experiment to look at how the one person had certain numbers of colonies on their hands before and after different sort of treatments. So they basically followed through this well-described protocol. And they put a lot of organisms on, about 30,000 per mil. So what you're seeing here is the individual 30 students. Some of them had very, very high levels of organisms. Many of them had extremely low levels of organism. This is the after ozone, and this is after disinfection of the hands with alcohol-based hand gel. So if you look at the people who are the very worst, the people who had the highest levels on their hands, those same people had extremely good reductions with the ozonated water. And that's why you have to not look only at the mean, you have to look at the individual people. So there are clearly some people who do hand rubbing very, very well, and so their counts are very low. So for those people, there's actually not much difference. But for the people who are pretty lazy or not very good at doing hand hygiene, you can see it makes a very, very large difference. So they concluded, that ozonated water was an effective way of decontaminating coliforms from the skin, and that might have some application in environments like nursing homes and even in public settings like cruise boats and supermarkets and lots of other places where people just need to wash their hands anyway, and there might be an argument for using something which is actually better, and those would be the reasons behind it. Now, interestingly enough, when they ranked the amount of irritation in the skin, about 1 in 5 of the nursing students got some burning or dryness from using the alcohol-based hand gel. And that wouldn't be that unusual in a sort of Scandinavian population. If they weren't using moisturizers properly, you would expect to see some level of hand irritation. If people use moisturizers properly, that background level should drop to be only 1 or 2%. However, the study was really neat because when the students used the ozonated water, none of them got irritation, and the author of the study, who was the ophthalmic surgeon, found that this is what allowed his dermatitis to resolve, and he got basically permission from the hospital to use that as his protocol, and that was what brought his skin health back so he could resume his surgical lists. So it's just a really neat illustration of the speed of the effect in a way that makes it easy to understand. Now, of course, when you use ozonated water, there is going to be a smell. And in this study, about 3/4 of the people who were there noticed the very characteristic smell of ozone gas. But when they measured using an ozone meter, I've got one of these at home. I've got various gas meters to measure different things, so it's pretty easy to measure. They found that the level was never more than 1/100th of a part per million. And the industrial exposure level that workers are allowed to be exposed of is actually 1/10th of a part per million continuously for 8 hours. You might be thinking, what on earth job would give you a high level of exposure to ozone? And the answer is if you're a welder or a boilermaker, because you spend a long time using a welder and you're often in an enclosed environment. And welding, of course, is throwing a very large electrical arc, so it does generate ozone. And in my former life as a farmer and working in engineering shops, basically doing welding all day, I can tell you from first-hand experience, you get a pretty good exposure to ozone, and that's where this level of 0.1 ppm actually comes from. It's an occupational health and safety level. So the reason I'm telling you that is that the transient smell that you get from the ozonated water, which lasts for a few seconds, is trivial. It has no problems associated with it from a workplace health and safety perspective. Now, the other point is that when they looked at the two different taps that they were using, one was a low flow tap. In other words, the water flow was less and the ozone concentration was stronger. When they looked at the high flow tap, the ozone concentration was lower, and that was the 0.8. So the reason I'm telling you this is that when you get an ozone tap installed and you then put an AWP meter in and you measure the amount of ozone in the water, it can be tweaked up or down by changing the water flow. If you turn the water flow down, the ozone level will go up because there's more time for the water to interact with the catalysts in the electrolysis process. If you turn the water flow rate up, there's less time for the water to interact with the electrodes, and therefore the level is going to be lower. So by adjusting the flow rate, you can actually tune the concentration. It's really as simple as that. I was talking with a specialist in Sydney about 2 weeks ago, and he'd done some measurements on his system, and he was asking me, how can I adjust this? It's pretty simple. If you want more, slow down the flow rate. Use a small valve to do that. So that's just a really neat thing from an installation point that you probably should know. There is other work about taking ozonated water and combining it with other types of disinfectants. In this study, they combine it with alcohol and with benzalkonium chloride to create a type of surgical hand preparation material. And some studies have looked at just using ozonated water just combined with regular liquid soap, and that's been shown to reduce the transient level of bacteria on the skin by over 97%. And you can do that by using an ATP luminometer with the luciferase readout to give you actually a quantitative measurement. So all this means that there is some potential applications for ozone water for hand hygiene, not only in a clinical setting, but actually in a more broader way, particularly in places like nursing homes and cruise boats, because this is where you find those really tough candidates like norovirus and rotavirus, and the spores, which are these little dense seeds, if you like, very resistant of organisms like Clostridium difficile. So if you've got an ozone tap installed in your surgery, as in this example shown over here, what would you possibly use it for in addition to the things I've already mentioned? Well, one of the obvious things to do is to use it as the source of feed water for your dental unit. Because ozone treatment sterilizes water, if you take the ozonated water and it's in a sealed container, you've basically got sterile water. So you're not dosing further bacteria into your dental chair. Now, if you let that water sit, obviously the ozone will go off, and as I've shown you already, depending on how many ions there are in the water, the half-life might be 15, 20 minutes, or it might be as long as 2 hours. But it's going to dissipate over time. However, if you take freshly ozonated water and you run that through your dental chair in a flushing action, you're then exposing the water lines to high levels of ozone, and that can break up the biofilm and give you disinfection of your water lines. So in essence, you're doing a type of shock treatment to your water lines. Now, the proviso that comes with this is that there isn't any natural latex rubber O-rings along the pathway, because they're actually quite sensitive to ozone in water. If you have normal industrial O-rings, they're not made out of latex rubber. They're made out of a nitrile-like material called a bromobutyl polymer, and they have no problems with ozone whatsoever. But natural latex rubber does have a problem. So there are some dental chairs where the manufacturers are a bit iffy about the ozone because they've used these much cheaper types of O-rings inside the dental chair. But most dental chairs will certainly tolerate it. It can be put as the feed water for your ultrasonic scaler. And if you use the water prepared fresh, then as you apply the ultrasonic energy, you actually enhance the disinfecting action of the ozone. And a number of periodontists have found that extremely useful because it raises the oxygen tension in the periodontal pocket. And that's great for getting rid of all the anaerobic bacteria that drive periodontitis and peri-implantitis. You can use it if you've got a particle jet device with an external tank. You can use it in exactly the same type of way. And if you've got an oral irrigator and using that to deliver some solution like an antimicrobial, like a cloxine or something like that, you can use the ozonated water to actually clean the system out, because there are lots of organisms like the Pseudomonas I mentioned before that will not be inactivated by chlorhexidine, and they can actually grow biofilms inside things like oral irrigators. That's why in my specialty of special needs dentistry, we never let patients bring oral irrigators in when they come in for cancer treatment, when they come in to have a bone marrow or stem cell transplant or things like that. A few other applications that have been developed include disinfecting dentures and impressions before things go off to the lab, and disinfecting study models when someone has taken something from the mouth and put it directly on top of a study model. They've then contaminated the surface of the study model. And it's quite difficult to decontaminate a study model without somehow ruining the integrity of the surface of the study model. But ozone water, because it works very quickly and it works from the surface, is actually a pretty good way of doing that. So I've just included a couple of relevant references there. If you want to dig into the literature around those topics, there are further references that I haven't mentioned in tonight's presentation, and they're all listed in this recently published, it came out in May of this year, article looking at the most recent evidence around the different ways that ozonated water can be used in a dental practice. And also look for that fact sheet on ozonated water, which is in the ADA's COVID-19 microsite as one of the resource documents. It was only posted very recently, so it'll be up towards the top of the ones that have been newly added on there. So that's the little presentation just to sort of take you through the science, and now we can have some time for some questions and answers. Excuse me, I'll just...
Hey, Laurie, there was a question along the chat room before, just saying, when you're using ozone, ozonated water or aqueous ozone as a hand washing product, do you have to use soap as well? Now, I know you sort of covered that a bit, but what's your thoughts on that?
Yeah, so you need the soap because you need something that can actually break up bioburden on your skin, and you need something which brings the water into intimate contact with your skin. So remember that soap is not just there to break up things like breaking up food when you put, plates in a dishwasher. It's also a wetting agent. So you do need to use liquid soap when you're doing your regular hand washing. But if you're using ozonated water, you're getting a really good disinfection of your skin as well, but in a way that's quite chemically mild to the skin. So the short answer to the question, Jason, is definitely yes. You do need to use your liquid soap. The ozone is not going to somehow magically get into a very low contact angle with your skin, because your skin, first thing in the morning, is naturally covered by lipids. So you notice this when you come out of the shower, the water beads on the surface of your skin. So one of the jobs of the liquid soap is to actually lower the surface tension and allow it to wet the surface of the skin more broadly. Of course, you just need to make sure you rinse all that detergent and soap off extremely well. Yeah, that's a really good question though. Yeah, thanks, Jason. Okay, Andrea said, I've always wanted to learn how to weld. Yeah, it sounds really exciting, but it gets a bit boring after a while. And if you're not very careful with your face shield, you can get a very, very bad thing called welder's flash, which is a type of sort of interim retinal damage, which isn't very good for people who weld, but it is a really cool thing to do. Okay, and then your second question, what about putting ozonated water in the ultrasonic with no detergent? Well, you wouldn't need detergent if you're putting it into your ultrasonic scaler. Most of the detergents that you've come across are high foaming detergents. So if you activate those, you get a huge amount of froth and bubbles, which actually is probably a bit sort of counterproductive in the setting of a subgingival irrigation environment. So you just use it to replace the regular water that you would have otherwise put into the bottle on the top of your ultrasonic scaler. And the really neat thing is, as the ozonated water flows through, it will actually break up and remove any biofilm that has been building up inside that system. Because while people often flush out and treat their water lines in their dental chair, they often forget they've got water lines in their benchtop scaler, their benchtop particle jet device, all these other bits and pieces where we've got tubing and water lines also grow biofilm. So running ozonated water through those is great from a decontamination point of view. Thanks for your questions, Andrea. They're really good ones.
And Laurie, just another thing. Recently, we went into a dental practice and we tested bug count coming out of the faucets on the tap, pre and post ozonated water, and we got some significant reduction in bug kill. We were quite surprised at the amount of bugs that actually come out of our tap water just straight out of the faucet. Tamara, my business partner, has a science background, and she conducted those tests, and the results were outstanding.
Yeah, it's actually surprising. Yeah, when we've done field tests in about 100 practices now, looking at not only the bacterial levels in the water coming from the dental chair, but also looking at the levels from the taps in the operatory. And it actually varies very spectacularly. And again, it's a lecture in its own right, but in a very simple term, if you're looking at just Brisbane, for example, we found variations within a suburb depending on the complexity of the street network of pipes. So as the suburb has grown, there have been some issues, and a few little basically dead ends have been created. If you look at a shopping centre, an older shopping centre tends to have much higher levels of bacteria in the water because it's often had more renovations, additions, bits and pieces, whereas a brand new shopping centre will generally have a much smoother pathway for the water through it. So you find that is another factor. And then when you move radially away from the CBD of Brisbane and you go beyond the western suburbs to Ipswich, you find the levels of bacteria go up. When you go to Toowoomba, they go further. When you go to Dalby, they go further. When you go to St George, they go further. I notice because we have clinics at St George and Dalby that we operate. And part of the reason for that relates to water hardness. So when you cross the Great Divide, when you go up the range, for want of a better word, you run into an area where the natural groundwater has picked up a lot of magnesium, calcium, and aluminium salts. And what these do, not only does it make it a pain to try to use shampoo and hand soap and things like that, the longevity of the chlorine added to the water is much poorer. So as the hardness goes up, the chlorine level goes down. And I did a really neat thing just before the COVID pandemic. I collected a water sample in every city that I was in over a year. And prior to COVID, I used to travel every week. So I've got samples basically over all of Australia, Southeast Asia. And we then looked at things like water hardness, chlorine concentration, etc. And the harder the water is, the lower the chlorine concentration was and the higher the levels of bacteria. So where you live makes a difference in terms of the region and where the water is drawn from. And then the design of the building you're in also makes a difference. The most graphic example I can give you about that is to look at the old dental school at Turbot Street before we moved to our new facility at Hurston, where we actually designed the water system properly. We took the water from about halfway up the street. The building beside us, the Brisbane Dental Hospital, took its water from the bottom of the street. So even though the two buildings were side by side, the points where the water was taken off the main were about 100 metres apart. And the flow dynamics were very different. So we basically took the dental school water from a high flow area, and the dental hospital took their water from a more stagnant area. And as a result, the ambient levels of water in the same building on the lower floor were different from those in the upper floor because there were two different water systems in the one building. So it was a really neat way of being able to show this action. So it's really important when you are doing waterline tests to look at your feed water, and ozonating your feed water gets rid of the bacteria. And that's why it's like the first principle is don't put rubbish in, because the organisms are just going to grow. So it's one of the most common things that people do with their water ozonator is they use it to put water into their chair. Now, you can then put into that a chemical additive to slow down bacterial growth. And there's dozens of those on the market and they work very well. But starting off with water with low bacterial counts is actually really important. So thanks, Jason, for mentioning that.
No dramas.
Really
mate, you can see the chats, but Michael's also asked if ozonated water can be used to disinfect hard surfaces like benches or dental chairs as a replacement to alcohol wipes.
Yeah, so alcohol wipes are generally not recommended by dental chair manufacturers to wipe over their dental chairs because of problems with pulling out plasticizers, and particularly from upholstery. Most of them actually say there are several recommended products which will be based on water with some alcohol, detergents, and normally a quaternary ammonium compound. So look at common products like, say, FD366 would be a really good example, or ViraClean. They're basically water and a quaternary ammonium compound that's also a detergent, and they've got reasonably broad spectrum activity. Those things tend to be fairly compatible with upholstery and with hard surfaces. Just a plain Alco wipe is actually not that good at disinfecting a surface. It was never designed for that. Alco wipes were designed for a quick wipe of the skin before you shove in a needle. So when we look at modern wipes, the technology of modern wipes is more than just alcohol. It's actually about the water and usually the quaternary ammonium disinfectant and surfactant. It's not really so much alcohol on a surface that's a rigid surface is actually a fixative, and it will tend to fix protein onto the surface. That's why when people used to use alcohol after using detergent, they always had to use detergent before to actually clean the surface, because an alcohol wipe doesn't really clean anything. So there is a little nuance about the alcohol in that particular sequence. But you can use ozonated water as a wipe down. That's one of the things that they do in some of those commercial settings that I showed you previously. So, yep, that's a really good question. Thanks, Jason. Okay, what other questions have we got on the chat that I can't see? Wondering about... Okay, did you have any recommended installers for ozonated soluble surgery? That's a question for you, Jason. That's not a question for me. Jason can sort you out. So O3 water there. What's the expression? Just wanted to check the length of time you would have to have a patient carry out pre-procedural mouth rinse. Okay, so pre-procedural mouth rinses, while in the ADA documents on COVID, we say it's always good if you can get a 20 or 30-second mouth rinse. When you actually time patients out, most patients will only do it for about 15 to 20 seconds. It turns out if you actually look at the speed of killing, then as you'd expect, and I showed this on some of the graphs, it's actually a logarithmic phenomena, which means that most of the organisms get killed in the first 4 or 5 seconds. That's when you lose the first half, and then you then take a half of the remaining half in the next 4 or 5 seconds. So by the time you get to around about 10 seconds, you're about 3 cycles of repeat in, which means you're down to about an eighth or a tenth of the original level. So you only need, I'm not telling you to shorten your rinse, but if someone can't rinse for very long and they don't get to 30 seconds, don't sweat it, because by 20 seconds, you're well and truly all over. The net benefit gain of the last 10 seconds is so small, it's actually difficult to measure unless you put a very high load in at the very beginning. A little bit like in the study I just showed you with the nursing students, they put a shellacking amount of the coliforms on their skin, far more than you would ever encounter normally, just to make it a really tough test. Yep, that's a really good question. Okay, do that. Do you need a special unit placed at the point of the feed water? So there are a couple of little scenarios here. Some water ozonators can be put downstream of a deionizing system. So if you want in your practice, deionized water in order to feed your steam sterilizer, for example, if you've got an instrument washer, a washer disinfector, then you would install one of those. So the mains water would go through that, and then you would basically then deliver off to those couple of things. You just basically split that out. So that's one way of configuring it. But many people using a kick tap just take the tap and literally just plumb it straight in. If you do put it through a deionizer first, then as I've shown you on those earlier slides from Paul Young's group, the half-life in water is much longer. So it goes from about 15 minutes up to about 2 hours. That's the benefit of going with the deionizer approach. So that's why I mentioned to you, so if you're aware of that, then you can decide what you want to do. But it would seem strange to just feed an ozonating tap with a deionizer. If you're going to put a deionizer in, you may as well use the deionized water for a few other things, like your steam sterilizer or your washer disinfector, or if you've just got an ultrasonic cleaner in your stereo room, use the deionized water for your final rinse, because it will have a much better end effect. You won't get all the residual things that cause spotting in your sterilizer chamber. So try to think about where you'd get the most benefit from. And somewhat bizarrely, I prefer the water deionizers to be mounted on the wall where you can see them. I know lots of people pop them into cupboards, but then sometimes people forget that every so many months you need to go in there and unscrew the cartridges and change them. Whereas if they're on the wall, they're just much more obvious. I know it looks ugly, but it's harder to forget when it's on the wall and easy to forget when it's in a cupboard. So they're just a few operational things that you need to think about. Good, that's a good question. Okay. Kath's been very, very mean. Time, 2-minute rinse. Well, good for you. Okay. Okay, what other questions have we got? How do we know the ozone machine is still working? So a couple of things. First of all, you should be able to smell it. If it comes out and you can't smell it, then Houston, you've got a problem, because as I said, the level that is generated in the ambient air, you'll be able to smell, and it's well above the detection limit for everybody. So if you can't smell it, then you've got a problem. You can get an AWP meter. I don't think it's a particularly good investment for you to buy an AWP meter, but you can get meters that will tell you how much ozone is dissolved in the water. Obviously, I've got one and we use it in our research, but if you really want to go that far, if you have a pH meter that's really good, you can buy an AWP electrode for a pH meter. But again, are you going to buy a $200 or $300 pH meter for that? Probably not. I've got a few. Would you buy them? Probably not. Okay. Hopefully that gives you a bit of a clue to that question. Okay. Can you help in the seminar to share with your staff? So this presentation is being recorded. So Jason will be able to make a link to it later on. So that way you can have your staff watch it afterwards. Jason's mentioned, yes, there is a monitoring piece of electronics that will tell you when the actual ozone generating bit, if you like, eventually needs a replacement when it's reached its mean time before it fails. So that gives you a little bit of an indication, so you can keep an eye on it by actually doing that. But it's a very long time. It's actually the hours of it running, not just the hours of it being switched on, because obviously unless there's water flowing through, the current is not doing the electrolysis. So many of these water ozonators last for an extremely long time. And now that you're a bit more aware of the technology, the next time you go past an ice machine, or if you're looking into a McDonald's or a Hungry Jack's or something like that, you see those large ice machines, you'll see a little water ozonator connected between the ice machine and the tap water fitting to make sure that the ice that's created isn't full of bacteria. Yeah, pretty obviously. Exactly the same sort of technology, but just arranged in a slightly different physical form for a device that has a fairly low and intermittent demand for water. I've got a couple of those, and I've tested them. They work extremely well. Really good. Excellent. Good. Do we have any other questions? Feel free to, you're all able to unmute yourself, so if you do want to actually talk, you can actually do that. So Andrea's now going to talk. That's good. Someone who's...
Okay, Laurie, if we don't have any other further questions, I would like to say thank you very much for your time tonight. Again, always, and as always, super informative, super impressive. I know that there's been a lot of interesting questions from the floor. And as I said, if anyone is chasing further information, Tamara and myself are more than happy to help. Our details are all over the invitations. But Laurie, I really want to say kudos to you. Thank you very much for your time. And I think we all enjoyed what you had to offer.
Great. No, thanks very much, Jason. And good evening, everyone. Have a really pleasant night. Thanks, everybody.
Cheers.
Okay.
Bye.
Bye-bye.